Good morning. This is Thursday, October 22nd. This is in regards to the retake of the experimental design test. A link for the test will be provided via your school email. You will have till Monday, October, let me check that date, October 26th until 5 p.m. to complete the retake. If you are going to do it online, you may also do it in person, on paper, after school, 12:45 on Friday, October 23rd, or Monday, October 26th. The time for the online test or the paper test is approximately 30 to 35 minutes. So let me walk through the previous test so maybe we can learn a little bit from our mistakes. So share my screen. So here's the last test. The test you will do for the retake has been changed. Same content, but different types of questions. So first question was about, do you understand the key vocabulary? Do you understand the key terminology? And so remember, a variable in general is any part of an experiment that can be altered or changed. And the two critical variables are the independent variable and the dependent variable. The independent variable is the variable you choose to change. It's the variable you are interested in exploring. It's the variable you are interested in testing. And so it's the one that is purposely changed by you, the experimenter. The dependent variable is the variable that you are choosing to watch, monitor, measure, so you know if your change in the independent variable made any difference. So as a researcher, you are manipulating the independent variable. And then how do you know if that manipulation of the independent variable makes any difference? You have to have a way to monitor or measure that. And that's where your dependent variable comes in. The dependent variable is the one that you're going to measure, monitor, observe, record, to see if your independent variable change made any difference. All other variables must become constant, which means they must be the same for every different experimental group to make sure that they are not influencing the outcome. So in one of the examples we used early in class, we talked about fertilizer in a cornfield. We were testing 3 different brands of fertilizer. So we were changing the fertilizer on purpose. That's what we were interested in exploring. So that's the independent variable. How are we going to know if the fertilizer made any difference? We identified that we would measure the height of the corn plant. And that was going to be the way that we could tell if the fertilizer was good or not. That means all other variables have to be the same for each plot of corn. So the corn receiving fertilizer X, the corn receiving fertilizer W, and the corn receiving fertilizer Z all have to have the same variables outside of the type of fertilizer. So same soil type, same amount of water, same temperature, same amount of sunlight, same type of seeds, all those things become constants. Otherwise, they could influence the outcome. Once one has design the experiment, they also want to make sure they have a control. A control is setting up a group that is used for comparison. Basically, a control group, you are not changing any of the variables. You're setting up the norm or the standard. So in the case of our corn field, we would set up a field of corn that received no fertilizer. Because if the field with no fertilizer outperforms the field with fertilizer, it tells you that it's not worth investing in the fertilizer. And the other term that you'll see in the list is hypotheses. Hypotheses is the educated prediction that you have for how your independent variable changes will affect your dependent variable. Question 2 talks about repeated trials. Doing something just once or twice, you're opening yourself up to error, you're opening yourself up to chance, you're opening up yourself to a fluke result. So the more trials you can get and the more consistent those trials become, the more weight, the more proof you have that what you're testing is actually valid. So in the research world, we want to see dozens to hundreds to thousands of trials. At a high school level, because of space, time, resources, we typically try to get 3 to 5 trials in out of practicality. But in reality, it would be much better if we could do 50, 100, 200 trials. That's going to make the odds that some fluke occurred or some random chance event happened, the data will sort of smooth out and eliminate those factors. In scenario 3, Gloria wanted to find out if the color of the food would affect whether kindergarten children would select it for lunch. She put food coloring into 4 identical bowls of mashed potatoes. The colors were green, red, yellow, and blue. Each child chose a scoop of potatoes of the color of their choice. Gloria did this experiment using 100 students. She recorded the number of students that chose each color. So what is Gloria manipulating? What is Gloria changing herself? What she's changing is the color that goes into the potatoes. She's the one that picked 4 different colors. She's the one that mixed the 4 different colors. So that's our independent variable, as it says, the color of the potatoes. And then how is she going to know if the color made a difference? She was counting the number of children that chose each color. So that becomes our dependent variable, the number of students that chose each color. What's not described above is a control group. In this case, a control group would be to set up a bowl of potatoes with no alterations, a bowl of potatoes with no changes, which means we'd be setting up a bowl of plain potatoes in their natural white color. And to see if students, because if most students pick the plain white potatoes, then we have no evidence that color is significant in the choice of food. Hopefully that makes some sense. Number 5, again, is getting at variables. There's a research question. Does the rate of crickets chirp change when the temperature changes? So what are we changing on purpose? Are we changing the rate of chirping or are we changing the temperature? Well, as the researcher, I really can't change the rate of chirping, but I can easily change the temperature. So the temperature is the one I'm going to change. That's my independent variable. And how do I know if the change in temperature makes any difference? I'm going to measure or count the frequency of chirps the crickets make in each different temperature. That's going to be my dependent variable. So you can see your best option then is B. The rate of chirping is our dependent variable and the temperature is the independent variable. C is also getting at the same idea, only this time we're looking for the constants. So we're looking at nitrate concentration and how it affects the size of the leaves. Again, as a researcher, I can certainly alter the amount of nitrate in the soil. As a researcher, I don't have any control on the size of the leaf. So what am I changing on purpose? I'm changing the nitrate concentration. That's my independent variable. And what am I going to measure to see if that change in concentration matters? I'm going to measure the size of the leaves. That's my dependent variable. So since nitrates is my independent variable and size of leaf is my dependent variable, those cannot be constants. Those are the 2 experimental variables. So those cannot be constants. So I'm looking for a choice that has something besides nitrate concentration and leaf size. So you can see A has nitrate concentration, that's eliminated. B has leaf size, that's eliminated. D has leaf size and nitrate concentration, that's totally eliminated. The only option left is C. The volume of water should be the same for all setups. Otherwise, water could be affecting your outcome. And the tree species that you are taking the leaves from and measuring should be the same. Each experiment should involve the same species to make sure that that is not an issue as well. So choice C makes the most sense. Number 7, we then gave you an experimental design and asked you to create a design diagram. And so you can read back through that. If you wish, you can pause the video and reread the description of the experiment. as you read the description of the experiment, we can see that what we're going to be doing is changing the pH. We are choosing to change the pH, so that's my independent variable. And we're going to measure the height of the bean plant in centimeters, as it says in the final sentence. That's our dependent variable. So we're going to use those two to make our title in our research question. So the title is the effects of acid rain, or you could have said the effects of pH of water, or the effects of the pH of soil. Any of those three would be acceptable, on the growth of bean plants, or you could have said on the height of bean plants. There's 1 point for the title. Notice we did include the species name, and we did include the period of the experiment, the duration of the experiment, which gives us a complete title. The effects of acid rain on the growth of bean plants, Phacelia vulgaris, over a 30-day period. You had to put the genus, species name, and the duration either in the title or the research question. It has to be in either the title or the research question, preferably in both. So the title, for this scoring, I gave the title 1 point or 2 points if you had the species name and the duration. Same with the research question. Will the pH, so I just have to turn that into a question. Will the pH of the soil, the pH of the water, affect the height of the bean plants, Phacelia vulgaris, over a 30-day growing period? So there's 1 point for the title, 1 point for the research question, and there was a third point between the two for including the genus, species, and the duration of the experiment. The null hypothesis, there's a typo there, it says Hall, it should say the null hypothesis. Remember, the null hypothesis states that what you're doing will have no effect. Null hypothesis states that what you're doing will have no effect. In research, we're trying to always reject the null hypothesis. We're trying to prove that wrong. So a null hypothesis would say a change in the pH of the soil will not affect the height of the bean plants, or something similar. But we're saying if we change the pH, it's not going to matter. All the beans are going to have approximately the same height. And we're going to try to prove that wrong in our experiment, which will then show that pH does matter. So your alternative states the opposite, states that pH does make a difference. So we just change the sentence to be the opposite. A change in the pH of the soil will affect the height of bean plants, or plants grown in different pHs will show different heights. Independent variable, we already identified, that's the one you're changing. Again, I would have taken pH of soil or pH of water. Levels are what different groups are you setting up. So in the description, we're setting up 4 different pots, and each one's getting a different pH. One pot is getting a pH of 7, another pot is getting a pH of 5, another pot is getting a pH of 3, and another pot is getting a pH of 1. If one of those is your control group, you should identify it in parentheses. So say pH 7 is the normal pH that we would use. If that's the normal pH, we could mark that as our control group. So remember, you should mark your control group if you have one. And the number of trials, in this case, we're measuring each seed separately. Since we're measuring each seed separately, each seed counts as a trial. So since we had 20 seeds in the pot, and we're going to measure all 20 seeds, that's 20 trials. So there'd be 1 point for the independent variable, 1 point for properly labeling your different groups, a second point for labeling the control, and then 1 point for properly identifying the number of trials. Your dependent variable was the height of the bean plant, and remember, you should always give the unit you're measuring in. So in parentheses, I put centimeters. So dependent variable, there's 2 points awarded, 1 for height of bean plant, and second point for identifying the unit you're measuring in, which is centimeters, in parentheses. Constants, it says, include a minimum of 5. These are all the different constants that were up in the paragraph above. So coming up with 5 should have been pretty easy. There was 3 points awarded for this part. If you had 5 of them and you had in parentheses, you defined what that was going to be, you would have gotten all 3 points. If you did not define them in parentheses, you may have only gotten 1 or 2 points. So don't just put type of pot, but give me a specification. It's going to be a clay pot. Don't just tell me the size of the pot, give me a specification. It's going to be 10 inches. Don't just tell me the amount of soil, tell me how much soil, 5 cups. Don't just tell me the amount of water, tell me how much water, 10 mils. Make sure that you define each of your constants when you list them. That means every pot, all 4 pots, this whole list has to be the same for all 4 pots. Otherwise, you can't be sure it's the pH that's causing the difference. Finally, there's 1 point for the predicted results. The predicted results says, what would you predict if you fail to reject the null hypothesis? Well, if we can't reject the null hypothesis, that means all the beans must have grown to approximately the same height, that there was no difference, there was no significant difference in the height of the beans. So if we ran our experiment in all 4 pots, the height was pretty much the same, we have no evidence to reject our null hypothesis. And it would appear that pH would have no effect. 2 improvements, that could vary. I've given 2 improvements here. I might set up more than 1 pot just in case the 1 pot gets a disease or a fungus or a mold or it actually gets knocked over. Good idea to have multiple pots set up. Also at the point, we had not identified a control group, which I just added brief moment ago. Then down below, we looked at the 4-question strategy, which we did in class with goldfish and we did in class with our bean project. Remember, all the materials that you can get represent possible independent variables. So these are all possible independent variables. And then these are all possible dependent variables, things about fish that you could actually monitor or measure. So the key is you had to take one thing from up here and match it with one thing from down here. So this would be your independent variable up here. This would be your dependent variable down here. And so your research question could be any combination. How does the size of the fish affect the number of offspring the fish produces? How does the amount of food affect the mass of the feces or poop? How does the, shape of the tank affect the pattern at which the fish swims? So there's literally several dozen combinations you could have done. Here's one example, not by no means the only example. How will the amount of food, right here, amount of food, given the tropical fish affect the mass of the feces, right here, mass. So I'm taking amount of food from up here, matching it with mass of feces down here. So how will the amount of food given to the tropical fish affect the mass of feces the fish produces? So I'm interested in knowing about feeding. I'm interested in knowing about how much food I should give my fish. And the way I'm going to know if the amount of food makes a difference is by measuring the mass of the feces the fish produces. My null hypothesis would say that what I'm testing will make no difference. So a simple statement, the amount of food fed to the tropical fish will have no effect on the amount of feces the fish produces. I expect that no matter what amount of food I give the fish, it's not going to make any difference in how much poop it makes. Again, I'm trying to reject that. I'm hoping to see there is a difference. But if I don't see a difference, my experiment's done. If I do see a difference in the amount of poop produced, then I go to my alternative hypothesis. The amount of food fed to tropical fish will affect the amount of feces the fish produces. I do expect to see a difference. But you can only consider your alternative if you first gather evidence to reject your null. Here's a second example. Size of fish tank, taken right here, size of fish tank, affect the rate of growth, right here from the second list. That's my dependent variable in tropical fish. Again, my null is going to say, it doesn't matter what tank you use, the rate of growth is going to be the same. The alternative says, if you put fish in different size tanks, there's going to be a difference in the growth rate. You first have to gather evidence to reject the null hypothesis before you can consider the alternative hypothesis. So that was the first version of the quiz. I'm walking through to hopefully help you do a better job on the retake, which again, that will become available later today, October 21st. You have until October 28th at 5 p.m. I will be sending a link for the online version, which is timed, and you only have 35 minutes in which to do it. Once you start it, you must finish it in one sitting, or it will lock you out. If you prefer to do it in person, it can be done in person after school at 12:45 on Friday, the 23rd, or Monday, the 26th. I think that pretty much does it. Thank you for your time.